All right. So good afternoon, everyone. My name is Temiloluwa Ogunyamoju. I am a student at Calder University, and over the summer, I have been at Vanderbilt University in the Spraggins Lab, which is a mass spectrometry research lab. And what I've been working on is investigating heterogeneity in human pancreatic tissue with multimodal imaging, such as the one that the lab uses. So I'm going to take you quickly through why we should care about the pancreas, the basic anatomy, lipids, the methods I use, results, and a bit of like my discussion and conclusions. So first off, I'm working with the pancreas. The pancreas is a largely important organ. It's a glandular organ, and it has two distinct functions. So it has the digestive function and the endocrine function. So we have the digestive function with pancreatic juices and enzymes that digest our food. We also have the endocrine function with hormones that help with regulating blood sugar, regulating our appetite, and so on. So as I said, we have two distinct portions, the endocrine and the digestive system, right? So the two main components of the pancreas are the exocrine tissue and the endocrine tissue. The endocrine tissue is more commonly referred to as the islets, which is what I have circled in black. So that is what it would look like with an H&E stain when you've imaged the tissue. So the one circled in black that's a bit lighter than all the other surrounding cells and tissues is the islets, and the rest is the exocrine. And then we also have some vasculature, but it's made up of really the exocrine and the islets. Now, looking at the basic anatomy of the pancreas, it is divided into three portions: the head, the body, and the tail. The head portion is connected to the small intestine, while the tail portion is connected to the spleen. Now, we have the islets scattered all across the pancreas, so in the head, the body, and the tail. But what we do know is that it is more abundant in the tail. But we don't know what the differences are aside that, or the diversity among like the head, the body, and the tail. So that is what I'm aiming to investigate. So the islet is really, it has a bunch of cells that secrete all the hormones that we need for appetite, blood sugar regulation, and so on and so forth. So I've spoken a little bit about the pancreas. Now I'm jumping into lipids. So I said I wanted to investigate heterogeneity in the pancreas, and I'm going to be comparing lipids in the pancreas to do that. Why? Well, because the multimodal imaging mass spectrometry that's used in the lab can provide us with that information, can help us analyze the lipids and help us identify the lipids within the pancreas. So I'm just going to take you through the basics of what a lipid is. So a lipid has hydrophilic head and hydrophobic tail, and it can be saturated tail or unsaturated. And the number of tails or the degree of unsaturation of salt, it contributes to the different classes of lipids that there is, such as phospholipids, triglycerides, and cholesterol, and so on and so forth, right? And all of these have important uses, such as in our biological membranes, right? Lipids are a very important component of the various biological membranes that separate our cell from its environment, or even the cellular interior. They act as messengers as well. So they're involved in a lot of pathways and internal mechanisms to help us do things such as secrete the hormones that the pancreas would secrete. And they also help define the cell structure and function. So that means that the lipid composition of different organelles can vary substantially. So they help define the cell structure and function. So we're going to be using lipids because of their importance in our cell and all of that, as well as because of the imaging technology that we have to investigate the heterogeneity that exists in the pancreas. So in regards to that, my project goal is to compare lipids in the head, the body, and the tail of the pancreas using multimodal imaging techniques such as microscopy and most notably multi-imaging mass spectrometry, which I'll take you through a bit later. So I'm going to investigate the heterogeneity or the diversity that exists within the head, the body, and the tail of the pancreas, and see if there is any differences that can be useful for us. So what is MALDI imaging mass spec? First, MALDI means matrix-assisted laser desorption ionization. Don't worry about it. I'm going to take you through what that basically means. But it is an imaging technique that allows the visualization of the spatial distribution of large biomolecules, for example, lipids that I spoke about, or proteins, or metabolites throughout a sample. So it basically shows how certain macromolecules are distributed in a particular tissue sample. So like, where are they located? What's their concentration in that part? So it visualizes the spatial distribution of those molecules. So how it works is you get a tissue section. So someone here had spoken with the kidney and how they section it. It's almost similar to how they section the pancreas. So I get a little block, and then I cut that, or I cryosection that into a very tiny piece, about 10 micrometers thick. And then I mount that on a slide, and then I apply a matrix, or like a coating. And what that matrix allows is for when the tissue sample goes into the machine that helps with generating the spectra, it helps the sample to be ionized way easier. So that's what the matrix is for. So we put it in the machine and we get the data. Lasers are shot all through the sample. And for each spot where lasers shoot, a spectra is collected at that spot of all the ions, the mass to charge ratio of the lipids that exist there. So that is basically a short on what MALDI IMS is. So now the basic method that I use for this project or this experiment is the lab had gotten the pancreas, I cryo-sectioned it, and then we went into microscopy, autofluorescence microscopy, then we went into the MALDI IMS process, sublimation, which is adding that coating or the matrix on top, and then put it into the machine to obtain the spectra at each laser shot. And then we did some more microscopy and then some staining as well. So I'm going to show you the imaging modalities I used and what the importance of those modalities were. So we have the before MALDI autofluorescence microscopy, which is basically for us to visualize how the tissue looks like, right? So visualize the tissue morphology, and it will help us determine what's the best area for us to image with MALDI. And then we have the MALDI IMS images, which, as I said, they help to determine the localization and the relative abundance of lipids within that region, right? So for example, in the image, the second image that you might be seeing on your screen, we have some yellow spots, we have purple, we have black. So all those are like ion concentrations, and they signify different things. So for example, the yellow are ions concentrated in the islet. We have the purple for the exocrine, and black for like vasculature, and so on and so forth. And then we have post-MALDI autofluorescence, which is after you've put your sample in the machine, and that just helps us to confirm the laser ablation, or like the laser shot size that we had set is the same. It also helps us to confirm the spatial resolution. Then we have H&E staining, and that just helps us to confirm and annotate some of the functional tissue units, such as the islets, the exocrine tissue, and the vasculature that exists. So I'm just going to show you pictures to kind of explain what it is I was looking for with this. So with the first image, it helps identify like all the units and just gives you a rough idea of where you'd want to image. And then with the post-MALDI autofluorescence, you can see like this white spots-ish in a row, in like in a grid pattern. So we then confirm the size of the ablation, or the size of the laser shot, and to make sure it's what we set. So we don't have overlapping of the shots, and which would mean we'd have overlapping of like the data we collected. So like collecting multiple data, right? And then we have the H&E staining, which helps us like visualize structures such as the islets a bit clearer. So now into some of the results of the experiment I was doing. So to determine like differences, one thing we first decided to do was get the measurements, the size of the islets, and see if that differs in the head, body, and tail region. So after we had imaged with the H&E and using HuBMAP, we were able to annotate the islets, and we were able to get this graph, which kind of shows that there's not too much of a difference, except one outlier in the body. There's not too much of a difference in the size. So in size, I mean like the area and the perimeter of the islets. So there was not a lot of difference there, is what we saw. And then the ion images, right? So this is an example of the ion image you would get from the MALDI process. So this ion, I chose ions that would help us annotate the islets in yellow, exocrine tissue and thereabout in purple, and the vasculature in black, right? So the box plot I have on the side is of one of the ions that's present here. And this ion was the most abundant ion in the spectra, in the average spectra. It was also the most abundant ion, or most localized in the islets. So it was really interesting. And a pattern that I kind of noted was that there was the median intensity for the box plots was higher in the body region. So for the graph, I apologize that there are no axes, but it's head, body, and tail as it is. So head, body, and tail. So the body in the middle, we can see a little bit like the median intensity is a bit higher. So that means the median intensity of that lipid is a bit higher in the body than it is in the head or tail. Another ion. So I went through, after getting the MALDI data, I kind of went through I went through a couple ions and I noticed some that seemed interesting. This is another ion that is localized in the islets. So you see the islets are like the bright yellow spots there. So there's another lipid that's localized in the islets. And we can also see a similar trend where the median intensity is higher in the body than it is in the head and the tail. Same thing for here. Here, visually, I was looking at this ion and I noted like the body, qualitatively, it looks like that lipid was reduced in the body, or was less intense or less abundant in the body. And looking at the box plot, you kind of do see that the intensity is a bit higher in the tail. You can also see that qualitatively, but also quantitatively, we can see because the median intensity of the tail islet is larger. And I had like a tentative identification of this lipid as well, which is interesting. This is in the exocrine tissue. So this lipid is localized in the exocrine tissue, and it seems to be least abundant in the body here, while it's more abundant in the tail, or like slightly on par in the head and the tail. It seems to be least abundant in the body of the pancreas. And for this one as well, this one is also a different lipid. Qualitatively or visually, I saw like, okay, it seems to be a bit more abundant in the body. And looking at the box plot, I was able to see that, right? So it's a bit more abundant in the body, as seen from the box plots. The median intensity in the body is greater than in the head or in the tail. So I've given you all that, right? So ongoing is, hopefully I have one more week of this internship. So hopefully I can get to do liquid chromatography tandem mass spec to further identify and quantify the lipids in each section. And future directions, what's the statistical analysis of these results? The differences that I'm seeing amongst like some of these lipids, are they statistically significant, right? Are those results statistically significant? If they are significant and we see that there's a difference in my results, can we then test other pancreas tissue samples from a diverse group of donors to see if that remains the same? So if those differences stay the same across a diverse sample set of healthy pancreases. and also like pairing the mass spec data with stains, such as we already did with the H&E, but to kind of identify the structures where the lipids are localized can also help us to identify what the lipids are and their structure and their importance, right? So I think I've spoken a little bit about this, and you might be wondering like, okay, what is really the significance of this project? So the big picture significance, I think this project is a small step in a long direction that could lead us to gold. But the big picture significance of this is, first, when we're comparing healthy or diseased pancreases, if we know that there's heterogeneity that exists within the pancreas itself in the different lipids that exist, we know when we're comparing healthy versus unhealthy pancreases, we know we have to compare the same regions, for example. And also, if we determine a particular lipid is more abundant in, say, the tail, and we find out that that lipid is important for maintaining cell structure or for secretions or something like that, and then we see that it's downregulated in a diseased pancreas, we can be like, oh, okay, what are some ways we can upregulate this lipid as a possible treatment mechanism or things of that sort, right? So this is there's a very large, broader picture to this research, but yes, identifying and quantifying the lipids in the different sections of the pancreas will probably allow us explore any heterogeneity present, thus generating knowledge that can aid in diagnostics, prediction, and prognosis in the future, enabling a more precise approach in the study and treatment of pancreatic diseases. All right, so thank you all so much. My acknowledgements, of course, the Spraggins Lab and all the members, my PI, my mentors, and every other person that trained me, onboarded me, HuBMAP as well for this opportunity, and all the mentors in HuBMAP as well as the NIH. I appreciate you guys for the opportunity. Thank you all so much for listening. This might have been a bit complex. I hope I was able to break it down. If you have any questions, I am more than happy to answer you there. You can just, if you noted the page number, you can let me know. But yes, thank you for listening.